Or can I see which one you're talking about? Okay, yeah, we were just talking about that. All right, so there's a question about mutation and base substitution, about the differences. So there's basically three different types of mutations. There's the missense, the nonsense, and the frame shift. So I'll go over each one real quick as soon as that turns on. Okay, so the first one is the base substitution mutation. It's also called a point mutation. Basically what it is a single nucleotide gets changed from what it's supposed to be to a different nucleotide. So in this case, if you look at these, you have these 3 A's followed by a C. That's the normal situation. For whatever reason, when it's being copied, usually there's a mistake. DNA polymerase adds the wrong nucleotide. And so this C that comes after these 3 A's accidentally got turned into a T. Okay? Now that gets transcribed to mRNA. That mRNA gets turned into protein. And when the ribosome's reading that mRNA to figure out what amino acid to put in that position, where this CCG gave a glycine originally, now it's TCG instead of CCG, and so it puts a serine there instead. So basically it's a single point mutation, a single nucleotide that causes a single amino acid to change somewhere in the protein. So a lot of times these are the least severe because it's just changing a single amino acid. Depending on how critical that amino acid is, it could be a big deal, but a lot of times it doesn't really change much. Okay? So this is the first time. It's called a missense mutation because it's just gives you the wrong amino acid.
So those two go by the same name, base mutation, missense?
Base mutation. A base mutation is a general term for a single base being
changed. Missense.
Specifically, this is a missense because it leads to just the wrong amino acid.
Okay. So you can still get the same amino acid?
You can, because of this, the way that the code is degenerate.
Okay.
So basically, let's say it was originally CGU, and it changed to CGC, you would still get an arginine.
Okay.
So that's a base substitution. The U got substituted for a C, but it didn't result in a missense mutation because the amino acid didn't change. Okay, does everybody see the difference there? The base substitution is at the nucleic acid level. The missense mutation is at the amino acid level. So you can get a nucleic acid changing from, again, in this example, if it were CGU and it changed to CGC, that's a base substitution because U changed to C, but when it gets converted to a protein, it's still going to be an arginine. So the overall product hasn't changed. Okay. So the other possibilities, there's the nonsense mutation. So again, this could happen from a single nucleotide changing. In this case, the fourth one was a T. It accidentally got changed to an A, and that caused the stop codon to be early. So basically, you get a truncated protein. The protein doesn't come to completion because it stops wherever that mutation was. That's a nonsense mutation. And again, the key there is that whatever the mutation is results in a nonsense codon, one of the 3 nonsense codons. So one of the UAA, UAG, or UGA. The last one is a frame shift. So instead of one base changing to a different base, that's basically a base just being removed. Okay, so if a base just gets removed, everything shifts over one. So basically, there's a mutation from that point on. Every amino acid past that has the possibility of being altered. Right? So here, again, you have these 3 A's in the middle. AAA. Let's say one of those A's just gets copied and gets removed. So everything shifts down by one base. And now instead of AAA, this C has moved into the third position here. So now it becomes AAC, and the next one becomes CGA. So everything is just shifted over by 3. And so every amino acid past that point has the possibility of being a mutation. Okay, that's a frame shift mutation. Okay, so those are the 3 possibilities for mutations. Okay. Anything else?
Can you explain the mouse again?
Mouse.
Yeah. Yeah, the experiment.
Phones keep ringing.
Okay. So I don't want to spend too much time on this, but I'll go over it briefly again. Okay, so you have couple scenarios here. So you have a bacteria, an encapsulated bacteria, that when it gets injected into a mouse, it kills the mouse. Okay? Let's not even worry about this for now. Okay, let's just worry about what happens to the mouse. Okay? So you take this living bacteria that's harmful to the mouse, you inject it in the mouse dies. Okay? Now you take a mutated version of this bacteria where the capsule, this bacteria doesn't have the ability to make that outer capsule, that protective capsule. And when you inject that into the mouse's immune system is able to kill that bacteria because it doesn't have this protective coating, and the mouse survives. Okay, so everybody see the difference between the first two scenarios? They're both living bacteria. It's just this bacteria has been mutated to not be able to make that outer protective coat. Okay. Here, if you take this first bacteria, but you kill it first, you heat treat it, so you kill the bacteria, okay, and then you inject that into a mouse, it doesn't kill the mouse, okay? Does everybody see the difference between 1 and 3? Outside. Nobody's phone in here?
Wow, I was lying. That's my alarm, I'm sorry.
Sorry. Guess we're not all going crazy collectively. All right. Anyway, so you have living encapsulated bacteria kills the mouse. You have a mutant bacteria without the capsule. The mouse's immune system is able to kill the bacteria. The mouse lives. You take this bacteria, which when it's living is able to kill the mouse. But if you kill the bacteria first and then inject it, the mouse survives, which would make sense because the bacteria is dead. Okay, so far it's pretty logical. Okay, the last one's the important one. So you take basically number 2 and number 3. So by themselves, neither one of these types of bacteria were able to kill the mouse. You put the two bacteria together and you incubate them together, and then you inject that mixture into the mouse, and the mouse dies. Okay. So by themselves, 2 and 3 weren't able to kill the mouse, but if you put them together and incubate them together, they kill the mouse. Now the reason is because this bacteria, although it's dead, has the DNA in it to make that protective coating. Okay? So when you kill it, it releases its DNA into the environment. That DNA can get taken up by this bacteria, which is still alive. It takes up the bacteria, it takes up the DNA from this bacteria and basically absorbs the gene to make that protective coating, and then starts to produce that protective coating. So basically, you now have some of this bacteria around because it's transformed, it's taken that genetic material from the dead bacteria, and now the living bacteria is able to make that protective coating. And if you take the blood from that mouse or the liver or whatever, and you plate the bacteria, you can see you do have this encapsulated bacteria around, even though what you injected was not living encapsulated bacteria. When you incubated them together, the genetic material got passed to the living bacteria. Okay, so this is a direct example of transformation, genetic material being passed horizontally. All right, I have one more question if there's anything else. Yeah.
I don't understand the repressor for induction and repression. The picture that you explained.
Show me when I get there. Tell me which one exactly you're talking about.
Oh, there.
Is there
Yeah, that's
this one? Okay. So repression you're okay with, but induction is a little bit fuzzy,
yeah.
or both.
Both, honestly.
All right. All right, so induction, the key thing about induction is in the normal state, this gene does not get transcribed, okay? So the cell doesn't transcribe this gene unless something in its environment tells it to. Okay, so there's a repressor protein which basically docks right downstream of where RNase polymerase has to dock and physically prevents RNA polymerase from transcribing that gene. Okay, so this is what it would look like in a normal situation. Okay? But let's say something in the environment does tell this cell, now we need to start transcribing this gene. So whatever that chemical is that's telling the cell to do that, it's called the inducer. It binds to the repressor and basically makes it fold differently to the point where it no longer can bind in the operator. Basically, it falls off the DNA. Now that it's fallen off, RNA polymerase can go through and transcribe the genes. So basically, it was physically in the way. Once it bound its chemical that tells it to start making that gene, it basically falls off the DNA. Okay, so that's induction. Again, normal state, it doesn't get made. In response to a stimulus, it does get made. Repression is the opposite. Repression in the normal state, it does get made. So in the normal state, which is shown here, the repressor protein doesn't bind the DNA. RNA polymerase is free to transcribe that gene. But when there's a stimulus telling the cell to stop transcribing those genes, it binds to the repressor. And again, opposite to what we saw, now when it gets bound to this chemical, it folds in such a way where now it does bind the DNA. And so now it's physically preventing the RNA polymerase from transcribing. So it's the exact opposite. Yep. All right. Anything else? We'll go one more if there's something. I think, Yvette, do you have something still? Or was it?
Just can you go over how the E. coli produces sugar toxin and
Yeah, so it would be the exact, that would be another example of the mouse experiment here. So basically, E. coli and Shigella was at some point in the same environment. Shigella released some of its DNA into the environment. E. coli took it up. And at some point, Shigella let go of its DNA that made the toxin that makes Shigella a toxic bacteria. E. coli took it up and started making that toxic protein. Okay, so it's just another example of transformation where one strain or one bacterial species passing genetic information to another one. All right.